Hello there, and welcome to the Sleep Porch. Tonight, we'll be drifting into the hidden realm of DNA, the tiny spirals of code that shape the natural world and make each and every one of us unique. DNA is life, written in a language so small that it hides at the very center of cells, and yet so vast that it carries the entire history of life on Earth. It is the microscopic engine behind growth, memory, healing, and every quiet transformation unfolding through the living world. Within this tiny molecular realm lie the foundations of creation itself. Instructions drift gently from one generation to the next, carried forward with astonishing precision, shaping life in ways so delicate and profound that they almost feel like whispers written into the fabric of existence. DNA weaves forms, builds structures, and holds mysteries that stretch back to the earliest dawn of biology. It is a world where chemistry becomes memory and where the smallest details guide the largest wonders. If you enjoy these quiet journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax, breathe deeply, let your shoulders soften, and allow your mind to gently unwind. Let's begin. A single gram of DNA could store more data than all human technology combined. If the entire digital output of humanity could be shrunk into a physical object, it would resemble a tiny speck of genetic material. The code of life is packed so efficiently that unimaginable amounts of information could, in theory, sit quietly in a space smaller than a sugar crystal. This is possible because each tiny unit along the molecule acts like a symbol, and these symbols can be arranged in near infinite combinations. Unlike digital machines, this form of storage does not require electricity or constant maintenance. When kept in stable conditions, the sequence can remain readable far beyond the lifespan of any modern device. Scientists have already experimented with placing texts, images, and simple moving scenes into these strands, then retrieving them with remarkable accuracy. The idea that living chemistry can double as a durable archive hints at future libraries that blend biology with information science, creating a bridge between the worlds of nature and technology. DNA is the most sophisticated information system known. Life uses a coding system that is both incredibly small and unbelievably powerful. Inside every cell, this molecular script guides how bodies grow, repair, and adapt. What makes it so extraordinary is not only its precision, but the way it integrates shape, timing, and instruction all in the same place. It holds long sequences that act like sentences, regions that behave like signals, and patterns that influence when its messages are used. Instead of simply storing details, it also builds the tiny machines that read and follow the very code it contains. The same set of instructions can tell a cell how to build the parts it needs, how to organize them, and how to respond when its environment changes. It operates continuously across countless cells without falling into confusion. Through all of this, the information remains stable enough to pass from generation to generation, yet still flexible enough to support new traits as life evolves. Every strand of DNA holds a story written in a 4-letter alphabet. Along each molecule sits a long line of repeating symbols that form the deepest biography any organism can have. These symbols appear simple at first glance, but their arrangement guides the creation of structures, pigments, enzymes, and everything else that keeps a living body working. Some segments determine how strong a muscle becomes, while others define how a cell reacts to a change in temperature or light. In many areas, groups of symbols work together to create rhythms, pauses, and prompts that help cells know when to begin a task or when to stop. Nestled between these regions are stretches that influence how quickly or slowly each message is used. Even the tiniest variation can alter the way a molecule folds or how a body responds to its surroundings. By reading these sequences, researchers can uncover ancient paths of inheritance and discover how traits have shifted over time. DNA can survive in conditions where almost nothing else endures. Long after soft tissues decay and bones crumble, small fragments of genetic material can remain protected in cold soils, dry caves, and other places where time moves slowly. The shape of the molecule, along with the bonds that hold it together, allows parts of it to resist breakdown for astonishing lengths of time. Even when heat, moisture, or natural radiation begins to wear it down, parts of the genetic script remain intact, just clear enough for scientists to read. These lingering fragments quietly reveal which plants or animals once lived in a place, how entire populations wandered, and what kinds of environments shaped their journey. In some of the planet's harshest corners, tiny organisms even carry repair systems that gently stitch their damaged genetic material back together, allowing them to endure conditions that would overwhelm most other life. Each surviving strand becomes a small message from the past, carried forward through time, waiting patiently to be discovered, waiting to be recovered and translated into a clearer picture of what once existed. The structure of DNA is a masterpiece of molecular architecture. Two long chains twist gently around one another, forming a shape that is both elegant and functional. This spiral arrangement makes the molecule strong enough to hold together through countless cell divisions, yet flexible enough to move and bend inside tight spaces. Along the inside of the spiral, pairs of matching symbols line up in a specific order, creating a stable pattern that can be copied with remarkable accuracy. The outside of the spiral is formed by repeating units that give the entire structure support, like the rails of a ladder. This design is not just visually appealing, it allows certain proteins and enzymes to find the exact places they need to interact with, without disturbing the entire molecule. When it comes time to copy the information, the spiral unwinds so that each chain can guide the creation of a partner. The entire form reflects a perfect balance between beauty, strength, and precision. DNA folds into forms far more complex than the famous double helix. Inside the cell, genetic material does not remain as a simple twist. It wraps around small disc-shaped proteins that act like spools, forming beads in a repeating pattern. These beads then link together and fold into thicker fibers, which in turn loop and curl into even larger shapes that fill the nucleus. This folding is carefully organized. Some sections are tucked away because their instructions are rarely needed, while others remain more open so that the cell can reach them quickly. Special helper proteins act like anchors, holding certain loops in place and creating neighborhoods where related instructions gather. All of this folding changes depending on the type of cell and its current needs. A nerve cell will arrange its loops differently than a skin cell, which helps each one specialize. By shaping itself in this intricate way, the molecule ensures that the right instructions are available at the right moment. The instructions in DNA can be read in multiple, overlapping ways. A single region can carry more than one message, depending on how the cell begins reading it. Sometimes, a shift in the starting point creates a completely different product. In other cases, the cell takes the initial transcript and rearranges certain segments before using it, creating new outcomes from the same sequence. There are also parts of the genome that contain hidden signals inside longer stretches. These signals control when a region is active, even though they do not contribute to the protein that is produced. Some sequences help regulate the speed at which the message is read, while others influence how the final molecule folds. This multi-layered approach allows the genome to conserve space while still supporting a wide variety of functions. Instead of assigning each instruction to a separate area, life weaves multiple uses into the same piece of code, creating an astonishing level of efficiency and complexity. DNA repairs itself with astonishing accuracy. Cells constantly monitor their genetic material for bumps, bends, or misplaced units that signal damage. When something looks wrong, teams of specialized enzymes move in. Some pry apart the affected region and remove the flawed section, while others rebuild the missing part using the undamaged chain as a guide. Additional enzymes reconnect the backbone so the molecule becomes whole again. During replication, proofreading systems check the new copy to ensure that no incorrect symbols slip through. When breaks occur on both sides of a strand, cells can either fuse the ends back together or rebuild the missing stretch by copying from an intact region. These systems work continuously, making rapid decisions about how to fix each problem. This constant attention keeps the genome stable over long periods, even though it is exposed to many forms of stress. Without this repair work, cells would quickly accumulate harmful changes that could disrupt their most basic functions. Evolution constantly rewrites DNA. Across many generations, small adjustments appear in the sequences passed from parents to offspring. Some arise when a copying error slips past the proofreading systems, while others come from environmental influences that nudge the molecule into a slightly different shape. Most of these changes have little effect, but a few alter the way a protein functions or shift the activity of a regulatory region. When a change offers an advantage in a particular environment, it spreads more easily. Over time, these shifts accumulate, giving rise to new traits, new behaviors, and sometimes new species. Portions of the genome can also be duplicated, providing extra copies that can explore new roles, while the original version continues its essential task. Other pieces move around, switch orientation, or become rearranged in surprising ways. All of these alterations turn the genome into a living record of adaptation, shaped by countless forces acting across long stretches of time. Your DNA contains echoes of ancient relatives you have never heard of. Within your genome are small segments that can be traced back to populations that once lived in isolated regions far from the places where their descendants now reside. As groups moved and encountered one another, pieces of their genetic material passed into new communities and were carried forward through the generations. Some of these segments reflect adaptations to local climates or food sources. Others preserve traits that were valuable long ago, but may be less noticeable today. Each piece acts like a quiet reminder of human history. By comparing these patterns across individuals, researchers can reconstruct migrations, cultural shifts, and moments of contact between groups that might otherwise be lost. The result is a molecular map of ancestry that reaches much farther back than written records. It reveals how interconnected human populations have been. Even when separated by oceans, mountains, or long distances, DNA can jump from one location to another in the genome. Some stretches of genetic material behave like tiny travelers. Instead of staying anchored in one position, they can move to a new location within the genome. These wandering segments shift their position through natural cellular processes, sometimes inserting themselves into regions they have never visited before. Their movement is not chaotic. Many of them use specific enzymes that help them leave one place and settle into another. When these wandering fragments finally settle, they gently reshape the landscape around them. They can awaken stretches of genetic code that once lay quiet, or soften the activity of regions that were once vibrant and busy. Sometimes, they even weave new combinations by carrying tiny bits of nearby material along for the ride. And while their movement may seem unpredictable, it has quietly fueled adaptation for millions of years, helping life evolve, adjust, and discover new possibilities, one subtle change at a time. Over enormous spans of time, these traveling sequences have helped shape genomes, adding variety and fresh possibilities that could not have emerged from simple copying alone. Their roaming behavior turns the genome into a dynamic landscape, constantly shifting in subtle, surprising ways. Genetic switches can control thousands of biological events throughout the genome, are small regulatory regions that act like switches, flipping instructions on or off depending on the needs of the cell. These switches do not encode structures or enzymes. Instead, they determine when certain messages are read, how strongly they are expressed, and how a developing organism responds to signals from within and outside the body. Some switches become active only under specific conditions, such as a sudden rise in temperature or a change in nutrient availability. Others work rhythmically, keeping time with daily biological cycles. A single switch can influence many unrelated processes by controlling a region that branches into multiple pathways. When several switches interact, the complexity increases dramatically, making possible the intricate patterns of growth and specialization seen across all forms of life. Even small changes in these regulatory zones can have wide-reaching effects, creating differences in shape, color, behavior, or physiology. They are a profound reminder that the genome is not just a list of parts, but a finely tuned system of timing and control. Silent regions of DNA hold secrets that shape our lives. Between the areas that actively guide the production of molecules, lie long stretches that appear quiet at first glance. These regions do not directly code for proteins, yet they play essential roles in directing how the genome functions. Some act as landing pads where regulatory proteins bind, influencing whether nearby sequences become active. Others provide structural support, helping the entire molecule fold into patterns that determine which sections are accessible. Some silent stretches serve as spacers that keep important regions at the proper distance from one another. They may also carry signals that affect how quickly a message is read or how readily a cell responds to developmental cues. While they were once dismissed as unimportant, ongoing research has revealed that many of these quiet areas influence traits ranging from immune responses to physical features. Even though they do not speak loudly, their presence shapes the flow of information in countless subtle ways. DNA forms loops and tangles to fit inside a tiny cell. A single molecule of genetic material is far longer than the cell that houses it, yet it fits comfortably thanks to a remarkable folding system. The molecule bends, curls, and loops into compact shapes that prevent it from becoming damaged or tangled in harmful ways. Some loops bring distant regions into close proximity so they can coordinate activity, while others separate conflicting areas to maintain order. Special helper proteins guide this folding process by holding certain sections together and loosening others as needed. The resulting structure changes depending on what the cell is doing. When a region must be read, that loop relaxes and opens. When the information isn't needed, the strand gently folds in on itself, becoming smaller and more compact, like a ribbon being softly gathered by careful hands. Each type of cell arranges these loops in its own unique way, shaping patterns that support its particular purpose. This quiet choreography allows vast amounts of information to work smoothly within an unbelievably tiny space. And remarkably, not all DNA rests inside the nucleus. Some of it is kept elsewhere, carrying its own quiet responsibilities. While most genetic material resides inside the central compartment of a cell, a smaller portion exists elsewhere. In tiny structures that produce energy, there are separate circular molecules that carry their own set of instructions. These instructions help maintain the machinery that powers cellular processes. This external genetic material is inherited along a different pathway than nuclear sequences, providing a parallel thread of ancestry that traces through many generations. Because it operates independently, it can evolve at a different pace, creating variations that may not appear in the main genome. These independent molecules also interact with nuclear genes, forming a cooperative system that keeps the cell functioning smoothly. Their existence reveals that genetic information can be distributed across multiple locations within the same organism, each contributing to essential tasks in its own way. DNA can be translated into sound and music. Researchers have found creative ways to convert sequences into audible patterns by assigning different tones to each symbol along the molecule. When played, these tones form melodies that reflect the underlying structure. Although the sounds themselves are artistic interpretations, they reveal patterns that might otherwise be overlooked when viewed only as text. Repeating stretches become rhythmic, rare combinations form unexpected notes, and long pauses create moments of quiet. Composers and scientists have collaborated to explore these patterns, transforming biological information into a new sensory experience. This translation does not change the science, but it provides a different perspective on the complexity and order within the genome. It demonstrates how information from living systems can cross boundaries into fields like music and design, inspiring curiosity and opening new ways of understanding the hidden architecture of life. You carry ancient viral DNA within your cells. Long ago, certain viruses inserted their genetic material into the genomes of the organisms they infected. Instead of being removed, many of these viral sequences became permanent residents, passed from parent to offspring through countless generations. Over time, most of them lost the ability to function as active viruses, yet their fragments remain scattered throughout the genome. In some cases, these sequences gained new purposes. They can influence nearby regions, participate in regulatory processes, or provide raw material for new adaptations. Their presence offers a glimpse into ancient encounters between species and the viruses that once circulated among them. By studying these remnants, scientists can trace old outbreaks, discover how viral elements shaped evolution, and learn how genomes defend themselves against new infections. These embedded sequences are records of battles long past, preserved within modern cells. DNA creates proteins using an elegant molecular assembly line. Within each cell, a sophisticated system reads instructions and converts them into working molecules. The process begins when a segment of the genome is copied into a temporary form that can be transported. This temporary copy travels to a tiny structure made of coordinated components that position each building block in the correct order. The growing chain folds as it emerges, forming complex shapes that determine its function. Each step is guided by specific interactions between the temporary copy and the building blocks it brings together. The entire system operates with remarkable accuracy, producing countless proteins that perform tasks ranging from carrying oxygen to breaking down nutrients. This assembly line works continuously, adjusting to the needs of the cell and responding to environmental cues. It transforms simple sequences of symbols into the diverse machinery that keeps life operating at every moment. Microorganisms can trade DNA like tiny biological messengers. In the microscopic world, some organisms share genetic material with surprising ease. They can extend small connections to pass segments of DNA directly to a neighbor, or they can release fragments into the environment where nearby cells may pick them up. Some even use viral light particles to transfer information between individuals. This exchange allows them to rapidly acquire new abilities, such as resistance to toxins, or the ability to use unfamiliar food sources. Because these transfers do not require traditional reproduction, they can spread through a community far more quickly than inherited changes. This rapid sharing creates dynamic populations that adapt in real time to shifting conditions. It also reveals how flexible genetic material can be, moving between organisms in ways that reshape entire ecosystems. For example, a sudden environmental change might trigger a small alteration in one region, while a different event leaves a mark elsewhere. Although these edits do not function like conscious memory, they create a record that researchers can later read. This approach opens possibilities for tracking how cells develop, respond to signals, or interact with their surroundings. It transforms the genome into an active logbook that captures the history of individual cells in a way no traditional tool could achieve. Some organisms rearrange their DNA during their lifetime. In certain species, genetic material does not remain fixed after birth. Instead, it undergoes deliberate restructuring as the organism matures. Some single-celled creatures break their genomes into many pieces, discard large portions, and stitch the remaining segments into a new configuration that suits their adult form. Other organisms rearrange specific regions to create new combinations of instructions that help them adapt to shifting environments. These changes are carefully controlled and rely on specialized enzymes that recognize particular patterns and cut or join them at precise locations. The result is a genome that evolves within a single lifetime, adjusting itself to new challenges rather than waiting for inherited mutations across many generations. This internal editing can influence feeding strategies, defense mechanisms, or developmental transitions. It reveals that genetic material is not always a static blueprint. In some forms of life, it behaves more like a flexible script that can be rewritten in response to immediate needs. DNA can be edited by nature with molecular scissors. Within living cells exist proteins that act like tiny tools, cutting specific regions of the genome with remarkable precision. These natural cutters recognize certain patterns and slice the molecule at those points, opening space for repair or alteration. The cell then uses different pathways to heal the break. Some pathways simply join the ends, while others insert new sequences or copy information from a similar region. Viruses, bacteria, plants, and animals all rely on these natural editing systems to defend themselves, adapt, and maintain healthy genomes. Some microbes use these cutters to eliminate invading genetic material, while plants and animals use them to remove sections that could cause harm if left unmodified. Over long stretches of evolutionary time, these editing events contribute to new traits and structural changes. The presence of such precise natural tools demonstrates how actively genomes are shaped by built-in mechanisms that manage, refine, and sometimes reinvent their own sequences. A single change in DNA can transform an entire species.
In rare circumstances, one adjustment in the long chain of symbols can shift the direction of evolution. This alteration might influence a protein that guides early development, prompting a change in the arrangement of limbs, the pattern of coloration, or the shape of a sensory organ. It might adjust a regulatory region that controls how often a gene is used, or under what conditions it becomes active. A seemingly modest difference at the molecular scale can create significant variations in anatomy or behavior. Over generations, these differences may become widespread if they offer an advantage in survival or reproduction. Eventually, they can help define a new branch on the tree of life. While most changes are minor, history shows that occasional single-step alterations can ripple outward, influencing ecosystems and reshaping entire lineages. This sensitivity demonstrates how closely large-scale biological diversity is tied to the smallest details of the genome. DNA holds instructions for making millions of different molecules. Each organism carries the capacity to create an extraordinary variety of molecular tools, using the instructions stored within its genome. Some instructions describe how to build enzymes that speed chemical reactions. Others specify structural components that give tissues their strength, flexibility, or color. Additionally, many sequences produce small regulatory molecules that help cells communicate and coordinate their activities. By combining different building blocks in countless arrangements, cells assemble molecules tailored to distinct tasks. The sheer number of possible combinations allows living systems to handle an enormous range of challenges, from processing nutrients to sensing environmental change. This diversity is not static. New molecular varieties arise as sequences shift and expand over generations, providing raw material for further adaptations. The capacity to produce such a wide range of molecules is one of the main reasons life can occupy environments as different as deep ocean vents, tropical forests, and frozen tundra. The shape of DNA influences how life unfolds. Although the sequence of symbols along the molecule is vital, the physical shape that the strand adopts also affects how instructions are used. In certain regions, the molecule bends sharply, creating pockets that attract specific proteins. Other areas twist into tighter spirals, making them less accessible unless the cell deliberately unwinds them. Some sections form small loops that bring distant regions into close proximity, allowing them to interact in ways that would be impossible if the molecule remained linear. These structural variations help determine which genes are active, how quickly messages are read, and how the genome responds to internal cues. Because each cell folds and arranges its DNA in its own gentle pattern, the very same sequence can lead to completely different outcomes, depending on the environment in which it rests. This soft, dynamic shaping gives every cell an added layer of control over its genetic material, allowing life to be more flexible, more responsive, and far more beautifully adaptable than it appears at first glance. Some species carry entire extra genomes hidden in their cells. A surprising number of organisms contain more than one complete set of genetic instructions. In addition to the main nuclear genome, they carry additional collections of sequences stored in separate cellular compartments. These extra genomes support specialized functions that the main genome alone could not perform efficiently. For instance, some plant species house independent sets of instructions within structures that govern photosynthesis, while certain protists maintain multiple genomic systems for different stages of their hour life cycle. These additional genomes often follow their own inheritance patterns, evolving at distinct rates
and accumulating unique combinations of traits. Their presence can enhance survival by giving the organism a wider set of tools to draw upon. This layered organization shows that genetic information can operate on multiple levels at once, with each genome contributing to a larger network of interactions that support growth, energy production, environmental adaptation. Your genome contains ancient duplications of long-lost genes. Over the vast timeline of life, regions of the genome have been copied and recopied. Some of these duplicate segments remain active, while others drift into obscurity. Even those no longer used can be detected as faint traces among the more recent sequences. These copies provide a record of past expansions where a gene was duplicated and allowed to explore new functions, while the original continued its established role. Some duplicates evolved into entirely different tools that shaped major transitions in evolution, such as new metabolic pathways or novel anatomical structures. Others have faded into near silence, leaving only small hints of their former activity. By examining these remnants, researchers can trace the history of genes that once played crucial roles in ancient organisms, but have since been replaced or transformed. They reveal how innovation often arises not from creating new sequences from nothing, but from copying what already exists and allowing variation to accumulate. DNA creates molecular machines that build every part of us inside each cell. Instructions are transformed into intricate structures that operate with precision. From these instructions, cells construct tiny machines capable of handling tasks as complex as moving nutrients, repairing damage, and generating energy. Each machine forms through a sequence of steps that links simple building blocks into long chains, which then bend and twist into the exact shape required for their roar. Some machines capture light, others transport ions, and many join into larger networks that guide communication across tissues. The accuracy with which these structures assemble is astonishing, especially considering the crowded environment within the cell. Minor adjustments in shape can change their abilities, and even small variations in the underlying instructions can create entirely new functions. Together, these machines perform the work that keeps living organisms growing, sensing, and adapting. They demonstrate how the information in the genome is not only descriptive, but deeply constructive. Epigenetic marks can change gene activity without offering DNA. Cells use chemical tags to label certain regions of the genome, guiding how actively each section is used. These tags attach to specific sites and influence whether nearby instructions remain open and ready or become tightly packed and quiet. The placement of these marks changes during development, shifting the identity of cells as they specialize. They also respond to signals such as nutrient availability, stress, and environmental conditions. Importantly, these modifications do not alter the underlying sequence. Instead, they work like adjustable settings that tune the behavior of genes. Some marks remain stable across cell divisions, allowing daughters to inherit the same patterns. Others are temporary, disappearing once the triggering event has passed. This system provides flexibility, enabling organisms to respond quickly without rewriting their genetic code. It adds an extra dimension to gene regulation, allowing the genome to behave differently under changing circumstances. DNA can act as its own chemical logic system. Within the genome are regions that interact in ways that resemble simple decision-making. Certain sequences become active only when multiple conditions are met, while others suppress activity unless specific molecules are present. These interactions arise from the shapes the strands adopt, the availability of helper proteins, and the presence of chemical tags.
When combined, they create pathways that respond to signals in a predictable manner. Some regions act like gates that open only when a particular combination of factors align. Others serve as checkpoints that prevent instructions from being used prematurely. This network of interactions allows cells to coordinate growth, adjust their behavior, and maintain stability. Rather than relying on a central controller, the genome uses local signals to produce organized responses. The result is a self-regulating system in which information and structure combine to guide complex patterns of activity. Cells read DNA using molecular motors powered by chemistry. Inside every living cell, small moving structures glide along the strands of genetic material, reading the sequence as they travel. These tiny motors use bursts of chemical energy to push themselves forward step by step, never losing their place, even in the crowded interior of the cell. As they move, they pull the strands apart just enough to expose the symbols they need to interpret. They then assemble a temporary copy of that region so the instructions can be followed elsewhere. Different motors specialize in different reading tasks. Some focus on copying entire stretches during cell division, while others read shorter passages to guide daily functions. Their speed, accuracy, and endurance are astonishing, especially considering how often they must navigate twists, loops, and shifting folds in the genetic material. Without these chemical engines, cells would not be able to access the information that keeps them alive. Their activity shows how dynamic the genome becomes once life begins to use it. Some DNA can fold into knots and form stranded structures. Although genetic material is often described as a simple spiral, certain regions can twist into much more unusual shapes. In places where symbols repeat many times, the strand can form tight knots that stabilize themselves through unique chemical interactions. In other regions, 4 separate stretches can come together to form a compact 4-stranded block that is sturdier than the usual double chain. These shapes appear and disappear depending on the environment inside the cell, the level of activity in the region, and the presence of specific helper proteins. When they form, they can influence how easily a motor can read that section, how often a gene is used, or how tightly the strand folds. Some of these shapes seem to help maintain stability, while others may act as road signs that slow down reading until the cell is ready. Their presence reveals that the genome contains far more structural diversity than the familiar double spiral suggests. DNA stores energy in ways that physicists are still trying to understand. Within the spiral of genetic material, small physical forces gather, shift, and release in patterns that are only partly explained. When the strand twists, bends, or tightens, energy becomes stored in those movements. This energy influences how the molecule behaves as enzymes approach, how quickly regions unwind, and how tightly certain loops remain coiled. Researchers studying these properties have found that even slight changes in shape can alter the distribution of tension along the strand. This tension affects which areas become more accessible and which remain sealed. The idea that a molecule containing instructions also carries physical energy that shapes its own behavior is deeply intriguing. It suggests that the genome is not just a passive script, but a working structure that uses physics to manage itself. As scientists study these forces, they uncover new ways in which mechanical properties influence biology, revealing layers of control that are rarely considered outside advanced research. Your genome contains remnants of ancient extinctions. Buried within your genetic material are faint signals from lineages that no longer exist. Long ago, populations rose, expanded, and vanished due to changing climates, migrations, and natural events. Before these groups disappeared, they passed a few fragments of their genetic identity into the ancestors of modern humans. Over time, these fragments mixed with others and became part of a broader mosaic. Many of these remnants no longer serve any clear purpose, yet they remain as quiet markers of long-forgotten communities. By comparing these signals among different people, scientists can map population patterns that have no written records. Some regions reflect groups that survive chromatic environmental changes. Others come from communities that met, exchanged genetic material, and then faded from history. These tiny echoes are rarer than well-known ancestral signatures and can only be detected with careful analysis. They provide a window into ancient worlds shaped by forces that left few visible traces. DNA communicates through chemical signals at lightning speed.
Inside the cell, activity along the genome is coordinated by countless signals that travel rapidly between molecules. These signals come from small chemical changes that happen when a region becomes active or inactive. When one part of the molecule opens for reading, nearby proteins can sense the shift almost instantly. They then pass the information to other proteins that adjust their behavior accordingly. This allows cells to coordinate complex responses within moments. If nutrients appear, if stress increases, or if a developmental transition begins, the genome can respond in a highly organized way through these fast-moving exchanges. Although these signals are not words or symbols, they carry information that shapes how the genome is used. Their speed makes it possible for even simple cells to adapt quickly to new conditions. The result is a communication system woven directly into the chemistry of life. Some life forms use alternate genetic codes. While most organisms interpret sequences with the same universal set of rules, certain species have evolved distinct systems for reading their instructions. In these organisms, symbols that normally represent one building block may represent another. These alternate rules can appear in unusual environments or in tiny organisms that have adapted to very specific lifestyles. Their genetic machinery has shifted over long periods of time, resulting in revised sets of assignments that still function smoothly. This reveals that the genetic code is flexible rather than rigid. Life can reinterpret its own symbols and still remain stable and functional. These alternate systems offer gentle hints about how the earliest genetic rules may have taken shape, and how living organisms can quietly experiment with new arrangements while still preserving balance and order. They remind us that the basic logic of biology holds far more variation and creativity than we once imagined. Within DNA itself lie delicate molecular clues, whispers from the first living cells on Earth, carried forward through time. Some of the oldest features of genetic material appear across all known forms of life, suggesting that they were present in the earliest organisms on the planet. These ancient sequences guide basic tasks such as reading instructions, copying the genome, and managing energy. Because so many species still use them, researchers believe that these features existed in the first simple cells that appeared billions of years ago. By comparing modern genomes, scientists can infer details about how early life stored information, maintained stability, and responded to primitive environments. Certain repeated patterns and conserved regions act like molecular fossils. They preserve traces of the earliest biological solutions
that allowed life to persist.
Studying these clues helps reveal how complex organisms eventually emerged from very simple beginnings, offering a rare look at life's earliest steps. Your DNA changes in tiny ways every single day. Inside each cell, the genome experiences small alterations caused by natural processes. Normal activities, such as the copying of the molecule or exposure to everyday chemicals, can nudge a symbol into a new form. Cells rapidly correct most of these shifts, but a few remain in place as harmless variations. Over the course of a lifetime, these small differences accumulate in different cells throughout the body. This means that the genetic material in one tissue may differ slightly from the material in another. Most of these changes are neutral, yet they contribute to the subtle variation that exists even within a single individual. This ongoing adjustment highlights how active and dynamic the genome truly is. It is not a fixed entity, but a constantly maintained and ever-changing system. DNA, in the dark depths of the ocean, works differently than DNA on land. Organisms living far below the surface face pressures, temperatures, and chemical environments that are unlike anything on land. Their genetic material operates under conditions that would disrupt the molecules of many other species. To function in these extreme surroundings, their genomes rely on specialized features that keep the strands stable and ensure that reading and copying proceed without error. Some deep-sea organisms have protective proteins that bind more tightly to their genetic material. Others use unique chemical adjustments that prevent the strands from unraveling. The instructions within their genomes also reflect the unusual challenges they face, guiding the production of molecules suited to cold, high-pressure environments. These adaptations show that the genome does not behave identically across all forms of life. It responds to the environment in ways that allow living systems to survive in places that seem almost impossible. The genetic code has mathematical patterns hidden within it. When researchers examine the arrangement of symbols and their assignments, they find repeating relationships that resemble mathematical structures. The code groups certain building blocks in ways that reduce the chance of harmful errors, creating patterns that increase resilience. Other relationships appear when scientists track how similar sequences behave across different species. These patterns are not obvious from a simple reading of the sequence, but emerge when large sets of data are analyzed. Some scientists believe these relationships reflect deep principles that guided the early development of life. Others think they arose through natural selection, acting on the need for efficiency and reliability. Whatever their origin, these patterns reveal that the genetic code is not a random collection of assignments. It possesses an internal structure that supports stability while allowing evolution to explore new possibilities. Some genes turn on only under extreme conditions. Within every genome are regions that remain quiet for most of an organism's life, waiting for very specific circumstances to awaken. Some of these genes activate only when temperatures climb past a certain threshold, helping cells protect themselves from heat stress. Others respond to unusually low oxygen levels or sudden changes in salinity. There are genes that switch on during drought, genes that activate when nutrients disappear, and genes that light up only in the presence of particular toxins. Their silence under normal conditions is not inactivity, it is preparation. These genes contain strategies for rare events that may occur only a few times in an organism's lifetime. When they finally turn on, they help stabilize the internal environment, route metabolic pathways, or trigger emergency responses that keep the organism alive. Their existence shows that genomes are built not only for everyday needs, but also for unlikely challenges that demand immediate adaptation. DNA can spiral into supercoils to prevent damage. Inside the cell, long strands of genetic material must stay compact, organized, and protected. One of the ways cells manage this is by twisting sections of the molecule into tight spirals known as supercoils. These spirals act like coiled ropes, storing tension that keeps the strands stable and prevents them from becoming tangled or stretched during routine activity. When enzymes need to read or copy a region, they adjust the tightness of these coils so that the section becomes accessible. When the task is finished, the coils tighten again to shield the area from harm. This constant adjustment relies on specialized proteins that add or remove twists, maintaining the ideal level of tension. Supercoiling also helps separate regions that need to remain quiet from those that are actively used, creating a natural boundary system within the genome. This protective strategy allows the molecule to function in a crowded environment without sustaining damage from mechanical stress. Certain plants store surprising genetic adaptations in their DNA. Some plants hold unusual sequences that allow them to thrive in environments that would be lethal to most species. In extremely salty soils, certain plants rely on genes that help pump excess salt out of their cells. In deserts, others carry instructions for producing specialized sugars that preserve moisture and stabilize their tissues in intense heat. Some plants in nutrient-poor regions store adaptations that let them break down minerals that few others can use. There are even species that have developed unique methods for capturing nitrogen in ways that differ from the familiar symbiotic partnerships seen in many roots. These adaptations are written into their genomes through long histories of environmental pressure and natural selection. Each sequence represents a solution to a specific ecological challenge. By studying these plants, scientists learn how life can invent entirely new strategies for survival, offering insights that may one day help agriculture thrive in shifting climates. DNA interacts with water molecules in intricate patterns. Even though genetic material is often discussed in terms of symbols and sequences, much of its behavior is shaped by how it interacts with the water that surrounds it. Every twist and curve of the molecule attracts a specific arrangement of water molecules that cling to its surface. These patterns shift gently as the genome folds, creating quiet layers of hydration that shape its flexibility and stability. Tiny water molecules slip into the grooves along the DNA strand, helping guiding proteins recognize certain regions and easing the way for sections to unwind. And when the surrounding environment grows more crowded, or the balance of salts begins to change, those hydration layers softly rearrange themselves, subtly reshaping the molecule as it adapts to its new conditions. This subtle dance between DNA and water affects everything from how instructions are accessed to how the genome responds to temperature. It reveals that the molecule does not exist in isolation. Its structure depends heavily on its immediate surroundings. Your DNA can reflect the environments your ancestors survived. Within your genome are regions shaped by the climates and challenges faced by ancestral populations. Some sequences evolved to support life in cold regions, helping the body regulate heat or manage energy storage. Others reflect adaptations to intense sunlight, influencing pigmentation and responses to ultraviolet exposure. There are regions connected to diets based on particular plants, grains, or marine foods, each influencing how the body processes nutrients. When groups migrated into new habitats, their genomes shifted subtly as individuals with helpful traits became more likely to pass those traits forward. These inherited adjustments do not dictate behavior or identity. They simply represent historical interactions between biology and environment. Today, they serve as molecular footprints, revealing the paths taken by earlier generations as they explored new landscapes and faced unfamiliar challenges. The genome contains enormous regions we still cannot fully explain. Despite decades of study, there are vast areas of the genome whose roles remain mysterious. These stretches do not encode proteins and do not behave like familiar regulatory zones. Some appear to influence how the molecule folds, while others may interact with proteins we have not yet identified. Certain regions are remarkably conserved across species, suggesting that they perform important tasks even though their exact functions are unclear. Others vary widely, hinting at roles that differ from organism to organism. As researchers develop new technologies to observe genetic material in motion, they uncover signs that these regions may help coordinate long-range interactions or stabilize sections needed during early R development. Their significance is supported by the fact that removing them often causes subtle but meaningful disruptions. These unexplained areas remind us that the genome still holds secrets even in its silent spaces. DNA creates molecular clocks inside every cell. Inside living cells, certain regions of genetic material contribute to internal timing systems that help organisms keep track of daily rhythms. These regions produce molecules that rise and fall in repeating cycles, guiding sleep patterns, feeding behavior, temperature regulation, and countless other processes. The instructions for these timing systems are stored directly in the genome, and their patterns are remarkably precise. When one part of the cycle increases, another decreases, forming a feedback loop that resets itself over and over. Light, temperature, and nutrients can adjust the pace, but the core rhythm remains internally generated. Different tissues maintain their own timing loops, but all coordinate through signals that keep the overall organism synchronized. These molecular clocks help cells prepare for regular changes in the environment, adjusting metabolism and activity in anticipation rather than reaction. This organization reveals how deeply biological timing is rooted in genetic instruction. Some bacteria carry genomes that rewrite themselves when attacked. In hostile environments, certain bacteria respond to threats by altering their own genetic material. When a harmful virus attempts to invade, these bacteria detect fragments of the viral sequence and store a copy inside their genome. Later, if the same virus returns, the bacteria use this stored fragment to guide defensive machinery that destroys the intruder. This built-in system acts like an adaptive memory, recording previous encounters and preparing for future attacks. As new viral threats appear, the genome grows more complex, accumulating records of past conflicts. This dynamic strategy gives bacteria remarkable resilience. They do not merely defend themselves, they update their defenses in real time, shaping their genomes according to the dangers they face. This flexibility demonstrates how even simple organisms can modify their information storage to survive rapidly evolving challenges. DNA can be used to reconstruct long-lost ecosystems. Scientists can extract genetic fragments from soil, ice, sediments, or ancient remains and use them to piece together the communities that once lived in a particular place. These fragments come from plants, animals, fungi, and microbes that left traces of their genetic material behind as they moved through the environment. Even when no bones or fossils remain, these molecular remnants persist long enough to reveal which species lived together, which predators hunted which prey, and how the landscape changed over time. By sequencing these fragments, researchers create detailed snapshots of forests, coastlines, or tundra that vanished thousands of years ago. This method allows scientists to explore ecosystems that were shaped by climates very different from today, offering clues about how species responded to shifting temperatures, rising seas, or expanding ice sheets. Each recovered sequence adds another thread to the tapestry of ancient life. Your mitochondrial DNA tells a different story than your nuclear DNA. Inside each cell are small, energy-producing structures that carry their own genetic material. This mitochondrial genome is much smaller than the nuclear genome, yet it preserves a separate record of ancestry. Because it is inherited through a different pathway, often tracing through maternal lines, it forms a continuous thread that can reveal migrations and population histories not visible in nuclear sequences. Mitochondrial DNA changes at a different pace, accumulating small variations that help track how groups spread across continents or became isolated in particular regions. It also contains instructions essential for energy production, making it a vital component of cellular function. When studied alongside nuclear DNA, it offers a second viewpoint on human history and biological evolution. The two systems together paint a richer picture than either could alone. DNA can move between species through natural processes. In the natural world, genetic material is far more mobile than it first appears. Certain microbes release small fragments of DNA into their surroundings, where other organisms can pick them up and weave them into their own genomes. Some viruses act as couriers, carrying bits of genetic information from one host to the next during the course of their life cycle. Even plants and animals can absorb genetic fragments from parasitic partners or symbiotic microbes under rare but well-documented conditions. These transfers are not accidents. They occur through physical contact, environmental exposure, or interactions with tiny organisms that routinely exchange material. When a fragment enters a new species, the receiving genome may treat it as a new tool, a new regulatory cue, or simply an extra stretch of sequence. Most fragments fade without effect, but a few influence traits in surprising ways, giving organisms abilities they did not have before. Over the long periods, this quiet movement of DNA creates unexpected links between distant branches of the tree of life. It is one of the ways evolution explores new possibilities, not by slow change alone, but through shared information passed across surprising boundaries. Some forms of life carry far more genes than humans. We often imagine ourselves as among the most genetically complex creatures on Earth. Yet many species quietly hold far larger libraries of instructions. Certain plants, for example, have experienced multiple rounds of genome duplication, giving them vast collections of genes that help them respond quickly to shifting weather, hungry pests, and changes in the soil around them. Their genomes are like deep, layered forests of information, quietly adapting, adjusting, and enduring in ways we rarely notice. Certain amphibians and fish carry enormous genomes filled with extra copies of regulatory sequences, growth factors, and metabolic pathways. Even some single-celled organisms maintain surprisingly large gene sets that allow them to shift lifestyles depending on chemical conditions around them. These expanded gene collections do not make these organisms more intelligent. They simply reveal different strategies for survival. Some species rely on chemical diversity, others on flexible metabolism, and still others on rapid environmental sensing. The variety of gene counts across life shows that evolution has no single blueprint. It uses whatever combination of sequences helps an organism succeed in its habitat. Whether that means a compact genome or one that stretches across millions of bases. DNA can be preserved in ice for incredible lengths of time.
In frozen environments where temperatures remain consistently low and biological activity slows almost to a halt, fragments of DNA can persist long after the bodies that produced them have vanished. Snow builds into compact layers of ice, and within these layers, tiny pieces of genetic material become sealed away from heat, light, and oxygen. These protective conditions limit the chemical reactions that normally damage DNA, allowing sequences to remain readable for far longer than they would elsewhere. Scientists have recovered material from ancient cores drilled deep into glaciers, as well as from the frozen remains of animals that roamed landscapes that no longer exist. Each fragment carries clues about organisms that shaped past ecosystems, including plants, microbes, and animals that have left no other trace. These preserved sequences enable researchers to reconstruct ancient communities, track changes in climate, and understand how species responded to environmental swings that unfolded over many thousands of years. The ice becomes a molecular time capsule, storing delicate information far beyond the lifespan of any creature. Your genome contains genetic fossils from early life. Hidden within the long continuum of your genome are faint relics from biological worlds that predate complex animals. These relics come from early viruses, ancient single-celled ancestors, and primitive molecular systems that were once essential to life on a young planet. Over immense spans of time, many of these sequences lost their original purpose. Mutations accumulated, active regions fell silent, and recognizable functions faded away. Yet even in their quiet state, these fragments preserve the outlines of early biological innovations. Some resemble parts of ancient enzymes used by the first cells to manage energy. Others look like remnants of early genetic elements that moved freely through ancestral populations. By reading these fossils, researchers reconstruct episodes in evolution that have no remaining physical traces. They reveal the deep origins of key biological processes, offering a rare view of how life built its earliest biochemical tools before spreading into the diverse forms we see today. DNA can coil into nanostructures smaller than any machine we build. The genetic material in a cell is not limited to simple spirals or loose strands. In certain conditions, parts of the molecule fold into incredibly small shapes that operate at scales far beyond current human engineering. These shapes arise when repeated sequences interact with each other, or when helper proteins encourage the strand to bend into compact forms. Some regions curl into tight rings. Others form short branching structures or tiny loops that stabilize sensitive parts of the genome. These forms create a landscape of nanocal architecture that guides how the molecule moves, how it fits inside restricted spaces, and how enzymes approach specific regions. The complexity of these shapes makes them difficult to witness directly, almost like patterns drawn in moonlight present, but hard to see. Yet with advanced imaging, scientists have begun to reveal a hidden world within the genome, where delicate structures fold and unfold as a cell moves from one state to another. These tiny nanostructures whisper a deeper truth. DNA is not only a keeper of instructions, but also a beautifully adaptable material, capable of arranging itself with remarkable mechanical grace. Some genes are older than the Earth's first multicellular creatures. Long before plants rooted into soil or animals swam through oceans, early microbes relied on a core set of genetic instructions to perform fundamental tasks. Many of these early instructions survive in modern genomes, preserved because they perform roles that remain essential. These ancient genes guide energy production, the copying of genetic material, and the management of key chemical reactions that support basic life. Their remarkable stability across billions of years indicates that nature discovered reliable solutions early on
and continued to rely on them even as life diversified into complex forms. When scientists compare these genes across distant species, they find similarities that stretch back to the earliest branches of the evolutionary tree. Studying them helps reveal what early life was capable of, how those first cells survived, and which innovations proved strong enough to endure through dramatic changes in climate, chemistry, and planetary conditions. DNA folding speed is one of the fastest processes in biology. Inside the nucleus, genetic material does not drift in long, loose threads. It must fold into orderly shapes almost immediately after it is produced. This folding happens astonishingly quickly, guided by helper proteins that pull, bend, and stabilize the strand with impressive efficiency. Within seconds, long stretches of DNA become packaged into structures that protect delicate regions and keep different instructions available in the correct order. This speed is vital for maintaining cellular health. Without rapid folding, the molecule would remain exposed to damaging forces or become tangled in ways that block important regions. Experiments that capture the process in real time show that folding proceeds with remarkable coordination, as if the molecule remembers the shape it needs to adopt. This swift organization is one of the reasons cells can divide, repair damage, and respond to signals without delay. It shows that the genome is as active physically as it is informationally. Some genes are activated only once in an entire lifetime. Among the many regions of a genome, there are instructions that come into play during a single, fleeting moment and then fall silent forever. Some guide early embryonic transitions that set the foundation for later development. They activate for a brief interval, perform their role, and then remain quiet for the rest of an organism's life. Others appear during the formation of specialized tissues, switching on just long enough to guide cells into their final identity. These short-lived bursts of activity require extraordinary precision. If they occur too early or too late, the entire developmental process can veer off course. The existence of such genes shows how the genome operates not simply as a list of available functions, but as a timed sequence of events. Each instruction has an exact moment in which it must act, allowing complex bodies to unfold from simple beginnings. DNA can trigger biochemical reactions simply by changing shape. The molecule is not a passive carrier of information. When a section bends, twists, or relaxes, it alters the chemical landscape around it. A sudden shift may expose a hidden area that attracts a specific enzyme, or tighten a region so that nearby reactions slow down. Some shapes create small pockets where regulatory molecules fit perfectly. Others present wide-open surfaces that encourage faster activity. These shape-driven influences allow the genome to guide biochemical processes without altering its sequence. Cells rely on these physical cues to decide which pathways to activate and how quickly to carry out essential tasks. In this way, the structure of DNA becomes a direct participant in cellular chemistry. It provides subtle signals that help coordinate complex reactions across the crowded space inside a cell. Your immune system rewrites DNA to defend you. Specialized immune cells carry genetic regions designed to rearrange themselves. During their development, these cells cut and reconnect parts of their DNA to create unique versions of defensive instructions. Each rearrangement allows the cell to recognize a different molecular pattern on potential invaders. By generating countless combinations through this controlled rewriting, the immune system builds an enormous library of possible responses. When a new pathogen appears, at least one immune cell is likely to carry a matching arrangement that can identify the threat. This capability allows the immune system
to confront unfamiliar microbes without needing to evolve new genes each time. The process is tightly regulated and limited to specific regions, so the rest of the genome remains stable. It is one of the most sophisticated defense strategies found in nature, allowing humans to survive in environments filled with constantly changing organisms. Some organisms store backup genetic instructions in unusual places. In many living systems, the nucleus is not the only location where vital information is kept. Certain organisms rely on clever secondary storage sites that hold extra sets of instructions for emergencies, regeneration, or major life transitions. Some single-celled species create tiny compartments that carry simplified versions of their genome, using them only during specific developmental phases.
There are insects that maintain backup instructions inside early embryonic tissues, allowing them to restart development under stressful conditions. Some plants protect reserve sequences deep within dormant buds, enabling entire branches or even whole individuals to regenerate after harsh winters or physical damage. In a few unusual microbes, spare instructions sit inside protective protein shells, shielded from toxins and radiation until conditions improve. These backups are not redundant copies of the same material. They often contain variations that offer flexibility during unpredictable environments. This hidden storage system adds a layer of resilience, revealing how life distributes information in creative ways to ensure survival across changing landscapes. DNA creates proteins that can fold in trillions of possible ways. When a protein is built inside a cell, it begins as a simple chain of building blocks arranged according to the instructions encoded in DNA. The moment it emerges, it starts shifting, bending, and curling until it settles into a unique three-dimensional structure. Each possible bend introduces a completely new shape, and each shape leads to a different function. The number of potential forms is beyond comprehension. Most shapes never occur because the chain naturally follows physical rules that guide it towards stable configurations. However, even within those limits, the range of possible outcomes is enormous. One sequence can fold into a structure that speeds up chemical reactions, while a slightly altered sequence produces a shape that carries signals or transports molecules. This ability to produce such a diverse array of tools from simple instructions is one of the reasons life can adapt so effectively. The genome acts not only as a script, but as a generator of intricate machines built from the smallest structural changes. Your genome contains hidden switches that can shape behavior. Scattered throughout the genome are small regulatory sites that influence when certain instructions are used. These sites act like tiny switches, responding to chemical cues, developmental stages, or environmental conditions. Many of them do not directly create any product. Instead, they adjust the activity of nearby regions that influence how organisms respond to light, sound, memory formation, or stress. Although behavior arises from complex networks within the brain, these switches help shape the underlying patterns that guide how signals are processed. Some switches adjust the sensitivity of neural pathways. Others influence how quickly certain messenger molecules appear or disappear. These regulatory regions vary from person to person, contributing to subtle differences in temperament, attention, or emotional responses. They do not determine destiny, but they help guide the way biological systems respond to experience. Their presence shows that behavior is shaped by both environment and deeply embedded control points that tune the activity of genetic instructions. Some genes appear to have no recognizable origin. Among the many regions of the genome, researchers occasionally find sequences that do not match any known relatives in other species.
These mysterious genes do not resemble the ancient conserved sequences found across life, nor do they match viral insertions or duplicated material from neighboring regions. They seem to have emerged from nowhere, appearing abruptly in a lineage without tier ancestry. Some may have evolved from stretches that once served no purpose, gradually gaining structure and function. Others may originate from rare events where unusual chemical changes create new patterns that natural selection can later refine. These orphan genes can influence traits unique to a species, from specialized forms of metabolism to distinctive reproductive strategies. Their existence challenges the idea that all genetic instructions must descend from ancient sequences. Instead, they show that new genetic solutions can arise independently, adding fresh innovations to the ongoing story of evolution. DNA reacts to light and heat in delightfully complex ways. Although DNA is often imagined as stable and inert, it responds to its environment in subtle and intricate patterns when exposed to certain wavelengths of light. Some regions shift their shape slightly, creating small structural adjustments that can influence how easily the molecule unwinds. Heat can cause parts of the strand to loosen while encouraging other regions to tighten. These changes do not rewrite the sequence itself, but they do affect how the genome behaves. Cells rely on these tiny reactions to detect temperature shifts, regulate activity during seasonal changes, or protect delicate regions from damage. In some organisms that experience dramatic environmental swings, these physical responses play an important role in survival. They help adjust the pace of biochemical activity. They help control when certain instructions are accessed and protect the molecule from being pushed beyond its safe limits whenever the environment becomes stressful. Through these quiet interactions, DNA reveals itself to be more than a chemical archive. It is a responsive, living material, shaped gently and continuously by its surroundings. Even within its sequence, the genetic code carries layers of redundancy, subtle safeguards built into its design that help life endure, adapt, and remain resilient across generations. When cells read the genome, they follow a set of rules that translate sequences into building blocks for proteins. These rules contain overlapping safeguards that reduce the chance of harmful mistakes. Multiple combinations of symbols often produce the same building block, allowing minor changes in a sequence to have no significant effect. Some regions are arranged so that small errors cause only gentle shifts rather than major disruptions. These built-in redundancies help maintain stability even when copying occurs under stressful conditions. They also allow evolution to explore new variations without risking catastrophic failure. By preserving meaning through alternative pathways, the code ensures that essential functions continue even when sequences drift over generations. This layered system shows that life values reliability as much as diversity. It also demonstrates that the genetic code is not a simple translation chart, but a carefully structured network of relationships that balance precision with flexibility. DNA may hold clues to forms of life we have not yet discovered. Within the known diversity of genetic sequences, there are patterns that hint at biological possibilities not yet seen on Earth today. Some repeated motifs appear to be the remains of ancient systems that have few living representatives. Although no direct evidence of extraterrestrial organisms has been found, the diversity within DNA reveals how flexible information storage can be. It encourages the idea that life elsewhere could follow different patterns while still relying on stable molecular systems to store and use information. Your cells carry DNA that reflects ancient migrations across the planet. Many regions of your genome trace back to long journeys taken by distant ancestors. When populations moved into new lands, they encountered unfamiliar climates, foods, and diseases. Those who adapted successfully passed forward sequences that helped their descendants thrive in those environments. Over time, these adaptations accumulated in different groups, forming patterns that reflect ancient paths across continents, coastlines, and mountain ranges. Some sequences highlight expansions into cold northern regions. Others point to crossings through deserts or along river valleys. When scientists compare these patterns across people today, they can reconstruct migrations that occurred long before written history. Each sequence does not tell a complete story by itself, but together they form a map of human movement shaped by chance, challenge, and exploration. These genetic footprints are quiet reminders of the many landscapes your ancestors once walked. Some organisms carry circular DNA that acts like a second genome. In addition to the main chromosomes stored in the nucleus, certain organisms maintain extracircular strands that operate independently. These loops contain instructions for specialized tasks from metabolism to stress resistance. In bacteria, circular DNA elements can carry genes that allow them to break down unusual chemicals or resist harmful substances. In some plants and fungi, circular strands provide instructions that support unique life cycle stages. Even in animals, small circular fragments appear under specific conditions, often emerging in tissues that experience rapid renewal. These loops replicate on their own and can increase or decrease in number depending on cellular needs. Their independence gives cells extra flexibility, allowing them to activate alternative pathways without altering the core genome. These circular elements act as auxiliary libraries, offering additional tools that help organisms survive unpredictable environments. DNA can influence how long molecules survive inside cells. Within each cell, messenger molecules carry instructions from DNA to the structures that build proteins. These messengers remain active only for limited periods before being broken down and replaced. The genome plays a direct role in controlling this timing. Specific patterns within a sequence determine how stable its messenger form will be, whether it lingers long enough to produce large amounts of a protein or fades quickly after a brief burst of activity. Cells adjust these patterns to fine-tune processes such as growth, stress responses, and metabolic rhythms. Some messengers degrade rapidly to prevent overproduction. Others remain stable to support sustained activity. This control system ensures that the flow of information matches the organism's needs at each moment. By managing the lifespan of these molecules, DNA helps maintain balance across countless biochemical pathways, coordinating timing with remarkable subtlety. Your genome contains passages that have not changed for eons. Across the long sweep of evolutionary history, some sections of the genome have remained almost perfectly preserved. These regions appear in many species that branched away from one another long ago. Yet their sequences differ only slightly. Their stability suggests that even tiny alterations could disrupt processes essential for survival. Many of these conserved passages help manage the earliest steps of development, guiding how cells divide and organize themselves. Others support fundamental chemical reactions that all living organisms rely upon. Because they remain unchanged across such deep time, these regions serve as molecular anchors, linking modern life to ancient ancestors. Researchers studying these sequences can trace evolutionary relationships with extraordinary precision, following shared patterns across mammals, birds, plants, and countless other lineages. Their presence reveals which genetic instructions have proven so effective that nature has preserved them through shifting climates, mass extinctions, and the diversification of entire ecosystems. They form a stable foundation beneath all the variation and novelty that evolution creates.
Some DNA replication methods are used only by exotic forms of life. While most familiar organisms copy their genetic material using well-known molecular steps, certain unusual species rely on methods that differ dramatically from the standard pattern. Some viruses copy their genomes using rolling mechanisms that cycle around circular strands. Other organisms break their chromosomes into many short fragments and replicate them individually before stitching them back together. In some deep-sea microbes, replication proceeds in bursts rather than smooth cycles, adapting to extreme temperature or chemical conditions around them. These alternative strategies quietly reflect the environments where these organisms first evolved, each one shaped by the pressures and possibilities of its ancient world. They remind us that life can build faithful, reliable copying systems from many different beginnings. And as scientists study these unconventional methods, they gain gentle glimpses into how the earliest forms of life might have carried out replication long before the familiar systems of today appeared. They also discover new variations in genetic chemistry that expand our understanding of what biological copying can look like across the planet. DNA can form tiny molecular springs. At small scales, the long strands of genetic material behave like flexible wires, capable of storing and releasing mechanical tension. When the molecule bends around cellular proteins or twists during reading and copying, certain regions behave like small springs that stretch and compress. These springs help distribute physical stress across the genome, preventing damage and guiding the movement of molecular motors that travel along the strands. The tension stored in these tiny coils influences how easily different regions open or close. Some springs loosen to allow access, while others tighten to protect delicate segments. Their action provides a crucial mechanical component to genetic regulation. Without these spring-like behaviors, the molecule would be far more vulnerable to breaks, tangles, and chemical instability. Their existence shows how deeply physical forces shape the behavior of genetic information, even at scales too small to see. Some organisms repair their DNA far better than humans. Among the many forms of life on Earth, a few stand out for their extraordinary ability to restore damaged genetic material. Certain bacteria survive intense radiation by breaking their genomes into pieces and then stitching them back together with remarkable accuracy. Some microscopic animals endure complete dehydration that shatters their DNA, yet repair it after rehydrating, returning to normal function without lasting harm. Certain plants exposed to high levels of natural chemicals or ultraviolet light use robust repair pathways that correct damage quickly. These abilities arise from specialized enzymes, unique genome structures, and tightly coordinated repair networks that outperform those found in humans. Studying these organisms reveals strategies for maintaining stability under extreme conditions. It also demonstrates that life has evolved many different solutions for preserving genetic information. Humans rely on one set of tools. Other species rely on far more resilient systems capable of surviving conditions that would overwhelm most forms of life. Your genome contains hidden regulatory landscapes. Beyond the familiar coding regions lie wide stretches of the genome that act as control territories, shaping how entire groups of genes behave. These landscapes contain clusters of small regulatory elements that work together, guiding when specific regions activate or fall silent. Some control heartbeat rhythms. Others influence muscle development, immune readiness, or metabolic balance. Their arrangements create long-range interactions, bringing distant segments together to coordinate shared tasks. Different tissues use different parts of these landscapes, which helps explain why the same genome can support many cell types with distinct functions. These regions are not easily identified because their effects often appear only when large sections are altered. However, when researchers map their activity, they uncover sweeping patterns that reveal how the genome manages complex processes. These landscapes operate like subtle conductors, ensuring that instructions unfold with proper timing and intensity. Some genes rarely mutate because they are essential for life. Across the genome are regions so crucial that they remain almost unchanged across large populations. Even slight alterations can disrupt vital processes, so natural selection removes harmful changes quickly. These essential genes often guide the basic steps of cell division, energy production, and structural organization. Because they must function correctly in every cell, the margin for error is extremely small. As generations pass, many individuals inherit identical or nearly identical versions of these sequences. This stability does not prevent evolution. Instead, it ensures that the fundamental machinery of life remains consistent, allowing other regions of the genome to explore variation more freely. When scientists study these rarely mutating genes across species, they find remarkable similarities that highlight their deep evolutionary roots. They form the molecular backbone on which more flexible systems can innovate. DNA can flip its orientation like a molecular mirror. Within some genomes are regions capable of reversing their orientation. These segments rotate around a central point, flipping so that the sequence runs in the opposite direction. Cells use specialized enzymes to manage this reversal, ensuring that the process occurs only when needed. When the orientation flips, nearby instructions can change their behavior dramatically. Some become active, others fall silent. In certain microbes, this mechanism helps switch surface features to evade detection by predators or immune systems. In other species, orientation flips control transitions between life stages or metabolic states. This reversible system adds flexibility without altering the sequence itself. It functions like a rotational switch that toggles between distinct biological modes. Such structural behavior shows that genetic regulation extends far beyond the order of symbols on the strand. It also involves the physical organization of the molecule in space. Some species carry genes that turn on only in darkness. In environments where light cycles shape survival, certain organisms have evolved genes that activate exclusively when illumination fades. Some deep-sea creatures rely on nocturnal instructions to manage metabolism during darker hours. Certain plants use night-active genes to process energy reserves after sunset, preparing for the next day. Some insects activate sensory instructions in darkness to navigate, avoid predators, or communicate through subtle signals. These light-sensitive patterns arise from molecular sensors that detect even slight changes in illumination. Once the sensors register darkness, they trigger cascades that awaken the appropriate genetic regions. Such systems demonstrate how tightly organisms connect their behavior to environmental rhythms. They also show that the genome functions as both a biological archive and a responsive network tuned to the cycles of the world outside.
DNA can be transcribed in both directions at once.
In some regions of the genome, two separate sets of instructions lie close together on opposite strands. When cells read these regions, they can produce two distinct messenger molecules from the same location, but in opposite directions. This arrangement allows a compact genome to support multiple functions without using additional space. It also creates opportunities for fine-tuning because the activation of one direction can influence the accessibility of the other. In some organisms, this bidirectional reading helps coordinate processes that must occur together. In others, it allows rapid shifts between two functional states. Although most of the genome follows a single direction at each site, these unusual regions highlight how densely information can be packed when space is limited. They reveal that reading direction is another dimension through which evolution can build complexity. Your genome contains tiny molecular timers that record development. Throughout early growth, certain regions of the genome activate in precise sequences that help guide the formation of tissues and organs. Embedded within these regions are small timing elements that track how far development has progressed. These elements influence when specific instructions become available and when they close again. They can delay activation, accelerated, or limited to narrow windows during which crucial transitions take place. As cells divide and specialize, these timers help ensure that each group follows the correct schedule. Their effects remain invisible in adults, yet the structures they shaped persist throughout life. By studying these timers, researchers gain insight into how complex bodies emerge from simple beginnings. They reveal that the genome does not merely store information, it also measures time. Some organisms can rewrite their entire genome when stressed. In certain living systems, extreme stress triggers an astonishing response that goes far beyond the usual adjustments of gene activity. Instead of simply activating or silencing a few regions, these organisms restructure the very architecture of their genomes. Under severe drought, toxic exposure, sudden temperature shifts, or starvation, specialized enzymes awaken and begin reshuffling large segments of DNA. They cut regions apart, move them to new positions, delete sections that no longer serve a purpose, and amplify others that help the organism endure the crisis. The result is an internal overhaul that produces a fresh genomic layout better suited to the harsh environment. This restructuring can create new patterns of gene activity, new metabolic options, and entirely new combinations of existing sequences. In some cases, the genome returns to its former arrangement once conditions improve, while in others, the new configuration becomes permanent, allowing future generations to inherit the altered design. This remarkable ability shows that evolution can operate on much shorter timescales than expected. It is not always a slow accumulation of small changes across countless generations. In these adaptable organisms, evolution can unfold inside a single lifetime through dramatic reshaping of genetic information. DNA influences how cells sense their environment. Cells never stop monitoring the chemical and physical conditions around them. This awareness does not originate solely from proteins or surface receptors. Much of it begins within the genome itself, where certain regions direct how cells construct the tools needed for environmental sensing. Some sections control the production of molecules that detect nutrients, while others guide the assembly of sensors that recognize temperature, acidity, or mechanical pressure. Additional regions influence how internal messaging pathways respond when those signals are detected. These genomic instructions help determine whether a cell will react quickly,
respond gradually, or ignore a fluctuation entirely. In organisms that experience strong environmental variation, cells rely on extra layers of genetic regulation to adjust their sensitivity, ensuring that they respond only when a change is significant. This tuning process can shift from one tissue to another, allowing different parts of the organism to interpret the same signal in unique ways.
Through these quiet mechanisms, DNA helps shape the relationship between living systems and the environments they move through, gently guiding adaptation, balance, and the coordinated responses that keep life in harmony with its surroundings. The genetic code itself holds surprising symmetries. When scientists look closely at the rules that translate its sequences into the building blocks of life, they uncover hidden patterns, subtle structures woven into the code that reveal a remarkable level of order tucked within something so ancient and small. Some groups of sequence combinations behave as mirror-like sets that balance each other, reducing the impact of errors that occur during copying or reading. Other relationships link patterns that appear unrelated at first glance, but share subtle similarities in chemical behavior. These symmetries help stabilize how instructions are interpreted, ensuring that minor variations do not disrupt essential processes. They also create a structured landscape through which evolutionary changes unfold. When a sequence shifts, it tends to move within these balanced patterns, making transitions smoother and less harmful. Some researchers believe these symmetries emerged from deep chemical principles that shaped the earliest stages of life, while others see them as a product of countless generations of natural selection. Whatever their origin, their presence shows that the genetic code is not a random mapping. It is an organized system with layered patterns that maintain reliability while allowing innovation to flourish. Some genes can silence entire regions of the genome. Within the genome lie genes whose products exert influence far beyond their immediate vicinity. These genes create molecules that travel to specific regions of DNA and initiate large-scale shutdowns of activity. They guide helper proteins to wrap wide stretches of the molecule into compact structures that block access to reading machinery. This silencing can extend across multiple neighboring genes, forming quiet zones that remain inactive for long periods. During development, these silencing molecules help define the identity of each cell type by shutting down instructions that would otherwise pull the cell toward a different fate. In adults, they maintain stable patterns that keep tissues functioning correctly. Some silencing events respond to environmental cues, protecting the organism from unnecessary or harmful activity under stressful conditions. The ability to silence entire regions at once allows cells to coordinate complex systems of regulation. It highlights the importance of managing not only what genes are expressed, but also which ones must remain undisturbed for proper biological balance. DNA can shape how cells communicate with one another. Communication between cells depends on a vast array of signals that travel through tissues and help coordinate countless processes. The genome influences every step of this communication network. Some regions specify the construction of signaling molecules that move between cells, guiding development, healing, or metabolic adjustments. Other regions shape the receptors that allow cells to interpret these signals. Additional sequences regulate how strongly a cell responds, how long the message persists, and which internal pathways it activates. This layered control makes communication flexible and adaptable, allowing organisms to maintain stability while still responding to change. In complex systems, such as immune responses or tissue repair, genomic instructions help synchronize large groups of cells, ensuring that actions unfold in the correct order. Without the influence of DNA on both signal production and signal interpretation, multicellular life would lack the coordination necessary to build tissues, maintain organs,
or regulate behavior. Your genome contains clues to how your brain develops. Long before a brain begins processing thoughts, its earliest structure is guided by networks of genetic instructions that shape its overall architecture. Certain regions of DNA influence how early cells divide into specialized groups. Others direct the growth of long pathways that connect distant parts of the developing nervous system. Some sequences help determine the balance between excitatory and inhibitory signals, while others affect how sensory regions take shape. These genomic influences do not dictate ports or personality. They form a foundation upon which experience builds. Subtle variations in these sequences contribute to differences in learning, memory, perception, and even resilience under stress.
By studying these areas, researchers uncover the timeline of brain formation and identify key transitions that occur before birth and during early life. These clues show how the genome provides the initial scaffolding for the most intricate biological structure known, shaping its layout long before its functions emerge. Some life forms carry genes that can be completely rearranged each generation. Certain organisms rely on a remarkable strategy to create continuous novelty. Instead of passing down a fixed arrangement of genetic instructions, they break apart specific regions during reproduction and reassemble them in entirely new orders. Each generation begins with a fresh configuration, often producing new traits and biological behaviors. This generational reshuffling can influence metabolic options, surface structures, or reproductive compatibility. The process follows precise molecular rules, ensuring that essential regions remain intact, while variable portions generate diversity. This strategy allows populations to adapt quickly to pressures from predators, parasites, or environmental changes. It also provides a constant source of innovation without relying solely on random mutation. For these organisms, genetic rearrangement is not an occasional accident. It is a built-in mechanism that ensures every new individual carries a unique combination of traits, shaped both by ancestry and by the restructuring that occurs during the creation of each generation. DNA can form molecular hinges that open and close. Within the long strands of genetic material are regions that behave like tiny mechanical joints. These hinges allow certain segments to fold inward or stretch outward, depending on the needs of the cell. When a hinge opens, it exposes instructions that were previously inaccessible. When it closes, it shields those instructions from enzymes that would otherwise activate them. The motion is controlled by interacting proteins that sense chemical conditions and adjust the hinge accordingly. This dynamic movement helps cells manage which regions of DNA are available at any given moment. It also contributes to the larger-scale folding patterns that shape the three-dimensional arrangement of the genome. These hinge-like structures show that DNA is not a static string. It is a responsive framework capable of precise physical transitions that influence genetic activity. Some genes only activate after many generations. Across long stretches of evolutionary time, certain genes remain silent until a specific combination of circumstances finally brings them to life. These genes pass quietly from parent to offspring, waiting for environmental conditions or genetic interactions that match their requirements. When they eventually activate, they can introduce traits that have not appeared in a lineage for countless generations. Some may help organisms adapt to unusual climates or new ecological challenges. Others may restore ancient capabilities that were once unnecessary but become beneficial again. This delayed activation shows how evolution preserves possibilities that may not be useful in the present, but could become vital in the future. These dormant genes expand the potential of a species by carrying silent instructions that may one day shape survival. DNA can fold to protect itself from radiation.
When harmful radiation drifts through living tissue, it can disturb the delicate architecture of genetic material. To soften this risk, the cell gently adjusts the way its DNA is arranged. Some regions curl inward, folding into compact, protective shapes that expose less of their surface to harm. Around the most vulnerable sections, extra proteins gather like a quiet shield, absorbing or deflecting the reactions that might otherwise leave a mark. Even in moments of stress, the cell moves with calm intention, reshaping itself to safeguard the fragile instructions it carries. In some organisms that live in high radiation environments, this protective folding reaches extraordinary efficiency, allowing their genomes to remain intact even under intense exposure. Once the radiation subsides, the protective configuration gradually relaxes, returning the molecule to a state in which normal activity can resume. This ability to change structure in response to external threats demonstrates that DNA plays an active role in its own defense. Your genome contains regions that act like molecular memory banks. Inside the genome are sections that store traces of past cellular events, allowing future processes to unfold with additional context. These regions record experiences by accumulating chemical marks, folding adjustments, and subtle alterations in accessibility. When a cell encounters stress, these areas store a record of the conditions, influencing how the cell responds if the same challenge appears again. In developing tissues, these molecular memories help ensure that early signals leave lasting impressions, guiding the direction of growth and specialization. Some memory regions influence immune readiness, while others track exposure to nutrients, temperature shifts, or developmental transitions. Although these changes do not alter the underlying sequence, they create persistent differences in how instructions are interpreted. Each mark acts as a reminder of a previous state, allowing cells to fine-tune their behavior over time. This internal memory system shows that the genome does not react to every moment as if it were entirely new. Instead, it carries subtle echoes of the past that improve stability, adaptability, and long-term coordination across tissues. Some species carry genes that let them survive extreme environments. Across Earth's most hostile habitats are organisms that thrive under conditions far beyond what typical life can handle. Their resilience is rooted in specialized genes that support survival under intense heat, freezing temperatures, crushing pressure, or high salinity. Some of these genes instruct cells to produce protective molecules that stabilize proteins at extreme temperatures. Others help maintain membrane structure when salt concentrations would normally disrupt cellular balance. Certain deep-sea organisms rely on genetic instructions that prevent damage at enormous pressures, allowing their internal chemistry to function normally miles below the surface. In deserts and frozen tundra, unique genes assist in water management, preventing dehydration or ice formation inside cells. These adaptations do not appear in a single location within the genome. They are scattered across multiple regions, forming networks that cooperate to maintain stability in environments that would destroy most life forms. Their existence demonstrates how genetic diversity equips organisms for survival in the planet's most challenging landscapes. DNA can trigger rare biochemical pathways with a single twist. The physical structure of DNA is an active participant in cellular chemistry, and a slight shift in its shape can unlock reactions that remain dormant under normal conditions. When a segment of DNA bends sharply, unwinds slightly, or tightens into an unusual configuration, nearby molecules sense the change and initiate new pathways. These pathways may produce protective compounds, activate emergency repair systems, or alter the pace of metabolic reactions. Such shaped-driven triggers help cells adapt quickly to sudden changes before slower genetic responses can take over.
In some organisms that face rapid fluctuations in temperature, acidity, or chemical stress, these shape-induced pathways can determine survival. They operate independently of sequence changes, relying entirely on the molecule's ability to store mechanical tension and release it in precisely coordinated ways. This capability reveals the subtle power of DNA as a structural signal, proving that information lies not only in the symbols, but also in the forms the molecule adopts. Some organisms use DNA to build crystalline structures. In a few remarkable species, DNA plays a structural role far outside its traditional function as an information carrier. These organisms use tightly packed strands of genetic material as the foundation for crystalline assemblies inside their cells. The strands align in repeating geometric patterns that create durable frameworks capable of withstanding mechanical stress. Some microorganisms use these structures to protect viral particles during transmission. Others rely on them for stability during dormant phases, forming crystalline shields that preserve essential molecules. The ordered packing of DNA in these cases surpasses the typical folding patterns found in most cells, creating rigid lattices that behave more like engineered materials than biological components. These crystalline arrangements show that DNA's chemical properties go beyond storage and copying. It can serve as a construction material when arranged. Others settle near the edges, where they fold into quiet structures that rarely open. The three-dimensional pattern formed by these positions influences which genes activate, how strongly they respond, and how quickly they can adjust to new conditions. Researchers have discovered that the folding arrangement can predict whether a cell will divide, specialize, or shift into a dormant state. In early embryos, distinct folding patterns appear before visible developmental changes, acting as molecular roadmaps that guide future growth. In adult tissues, disruptions in these patterns can correspond to stress responses or altered metabolic states. These predictive properties highlight the importance of structural organization as a layer of genetic regulation. The genome shape is as meaningful as its sequence. Some genes are inherited in highly unusual ways. Most genes follow standard inheritance patterns, but a small number move through generations in ways that defy typical rules. Some genes pass only through one parent, remaining inactive in the other until specific conditions awaken them. Others copy themselves more frequently than standard sequences, increasing their chances of appearing in offspring even when they offer no direct advantage. Certain genes remain silent for several generations before suddenly becoming active again. Some even move around within the genome before being passed forward, changing their location between parent and child. These unusual patterns arise from unique molecular mechanisms that evolved for reasons not always clear. They can influence traits, ovulation structure, and the rate at which new variations appear. Their behavior reveals that inheritance is far more diverse than the familiar patterns described in standard genetics. DNA helps cells keep track of their internal states. Cells must constantly monitor not only their environment, but also their own internal condition. DNA contributes to this process by guiding the production of molecules that report on energy levels, nutrient availability, and the progress of developmental stages. Some genomic regions activate only when a specific internal threshold is reached, such as a buildup of metabolic intermediates or a shortage of vital resources. These regions help cells adjust their behavior by signaling when to divide, when to repair damage, or when to conserve energy. In multicellular organisms, these internal cues coordinate with signals from neighboring cells,
creating a balanced response across tissues. The genome's ability to encode these internal monitoring systems shows that it functions as a central reference point for cellular identity, stability, and self-regulation. Some organisms carry genes that switch forms throughout their lives. In certain species, specific genes exist in multiple structural forms that shift as the organism develops. Early in life, a gene may appear in one configuration that supports growth or movement. Later, the same gene rearranges itself, producing a different version suited to reproduction, defense, or environmental adaptation. These shifts occur through controlled restructuring, guided by molecular machinery that senses developmental milestones. The transitions are not random. They follow predictable patterns that ensure each stage of life receives the instructions it needs. This ability to change form allows a single genetic region to support many roles without requiring separate sequences. It demonstrates the creative strategies evolution uses to maximize flexibility within a compact genome. DNA can be used as a template for building nanotechnology. Researchers have discovered that DNA strands can serve as precise scaffolds for constructing nanoscale devices. Because the molecule naturally pairs in predictable ways, scientists can design sequences that fold into shapes such as boxes, ladders, cages, and moving hinges. These engineered structures can hold tiny particles, arrange molecules into ordered patterns, or act as frameworks for miniature machines. By adjusting the sequence, researchers control the shape with remarkable accuracy, allowing the creation of devices much smaller than traditional materials can support. These nanocal constructions offer potential uses in medicine, chemistry, and advanced computing. They demonstrate that the stability and predictability of DNA make it an ideal building material at scales where conventional engineering becomes difficult. The genome holds keys to understanding how life first organized itself. Embedded within modern genetic material are clues to the processes that shaped the earliest living systems. Certain conserved sequences mirror the functions believed to be crucial in the first biological networks, such as copying information, managing energy, and building essential molecules. Other regions resemble ancient fragments that once helped early cells maintain stability in harsh environments. By comparing genomes across many species, scientists can trace which instructions appeared early and which emerged later. This reveals patterns showing how simple molecular systems gradually gained complexity, coordination, and structure. These insights help researchers piece together the progression from primitive chemistry to the organized networks that define life. The genome preserves these ancient signatures as part of its ongoing history, connecting modern organisms to the earliest steps of biological organization. DNA can shape the chemistry of entire ecosystems. In living communities, DNA does much more than define individual traits. It helps determine how entire ecosystems function by influencing the chemical processes that take place across different species. When genetic instructions shape the roots of plants, they determine how nutrients move through soil. When genetic instructions shape the digestive enzymes of animals, they quietly guide how organic matter moves through forests, grasslands, and oceans. In the soil and sea, tiny microbes use their own genomes to decide how minerals will be broken down and transformed into forms that plants and animals can absorb. Some living beings carry genes that let them fix nitrogen into the earth, enriching the soil with every breath they take. Others release subtle compounds that ripple through the water, shifting its chemistry in ways that support entire ecosystems. All of it happens softly, almost invisibly. yet it keeps the living world in motion.
Across these interactions, DNA sets the rules for how energy flows, how waste is recycled, and how chemical balances remain stable. Even small changes in the genomes of key species can alter entire environments, influencing everything from acidity to carbon storage. When scientists examine these patterns, they see how deeply genetic instructions shape the chemistry of life at every scale. DNA becomes a quiet architect behind forests, reefs, wetlands, and all the interconnected processes that allow an ecosystem to thrive. Some genes have survived virtually unchanged since the dawn of life. Among the countless sequences that fill modern genomes, a select group stands out for its remarkable stability across deep time. These genes appear in bacteria, plants, animals, and even single-celled organisms that branched apart billions of years ago. Despite the enormous diversity of life, these sequences remain nearly identical,
Suggesting that their roles are so essential that nature cannot afford to change them. Many of these enduring genes support fundamental processes such as energy transfer, molecular copying, or protein folding. Others help maintain the internal structure of cells or regulate reactions necessary for basic survival. Their persistence reveals the continuity between the earliest life forms and the organisms alive today. When researchers compare these sequences across species, they uncover a shared thread that connects modern biology to the first living systems on Earth. These genes function like ancient tools that have served life since its earliest chapters, unchanged because they work with unmatched reliability across every environment ever inhabited. DNA interacts with forces at the edge of quantum biology. At the smallest scales, the structure of DNA is influenced by physical forces that operate at the boundary between classical chemistry and quantum behavior.
Electrons within the molecule move in ways that follow quantum patterns, allowing them to shift positions between atoms in fractions of a second. These movements help stabilize the double helical structure and guide how bonds form and break during reading and copying. Some researchers propose that quantum effects may influence how DNA detects certain chemical changes or how it manages energy during replication. Although these ideas remain an active area of investigation, experiments show that the molecule responds to subtle fluctuations in the positions of electrons, suggesting a level of sensitivity far beyond ordinary chemical intuition. These interactions do not imply that DNA behaves like a quantum computer. Instead, they reveal that life operates in a space where tiny physical forces shape stability and precision. This intersection between biology and quantum physics offers new perspectives on how information can be stored with such reliability inside a molecule so small. Some organisms can pause genetic processes for astonishing lengths of time. Life does not always operate on continuous timelines. Some species possess the remarkable ability to halt key genetic activities when conditions become unfavorable. During droughts, extreme cold, or nutrient loss, certain organisms suspend the reading of their genes, allowing daily processes to slow almost to a standstill. While paused, their DNA remains intact but unused, protected inside stable cellular structures. This suspended state can last months, years, or even centuries in rare cases. When favorable conditions return, the paused genetic systems reawaken, reading instructions and rebuilding activity as if no time had passed. These organisms use protective molecules to stabilize their genomes during the pause, preventing damage from temperature fluctuations, oxidative stress, or radiation. Their ability to store living information in a dormant state demonstrates that life can stretch across time in ways that challenge ordinary biological expectations. It also reveals strategies that may offer clues to long-term survival in space or extreme environments on Earth. DNA can form molecular patterns that influence evolution. When genetic sequences are arranged in certain ways, they create patterns that shape the direction of evolutionary change. Some sequences repeat at regular intervals, forming structures that encourage variation through copying errors or rearrangements. Others cluster into loops that fold in predictable ways, guiding which regions are accessible for mutation. Certain genomes contain areas where mutation rates naturally run higher, acting as experimental zones where new traits can emerge. In contrast, other regions remain tightly protected, reserving essential instructions with remarkable stability. These patterns form a landscape that filters which changes are likely to appear and which remain rare. Over time, this landscape shapes the evolutionary path of the species, directing innovation toward some areas while shielding others from disruption. The patterns do not dictate outcomes. They create a map of possibilities that help determine how life explores new forms, new strategies, and new solutions to environmental challenges. Your genome contains deep molecular history beyond human origins. Within your DNA lie chapters of history that extend far beyond the emergence of humans. Some regions reflect ancient primate ancestors that lived in forests long before modern humans evolved. Other regions trace back to early mammals that survived dramatic shifts in climate and geological change. Still deeper sequences reach into the era of early vertebrates, capturing fragments of instructions that once guided the formation of fins, gills, and simple sensory systems. Even further back, faint molecular echoes reflect early multicellular life and single-celled ancestors that left no fossils. These molecular traces do not define personal identity, but they reveal the astonishing continuity between modern organisms and deep evolutionary past. Each sequence represents a solution that proved successful in earlier worlds and persisted across branching lineages. Your genome is not simply a record of recent ancestry. It is a layered archive containing molecular footprints from almost every stage of life's long journey across the planet. Some species share genetic solutions despite being entirely unrelated. Across Earth's diverse habitats, unrelated species often develop remarkably similar genetic strategies to solve similar problems. This phenomenon occurs when different organisms independently evolve sequences that perform equivalent functions, even though their last common ancestor lacked those features. Some desert plants and Arctic animals share genetic tools for managing dehydration or preventing cellular damage during freezing. Certain insects and marine organisms independently developed similar detoxification genes that help them survive environments rich in harmful chemicals. Even microbes living in distant locations sometimes evolve comparable sequences to handle shifts in acidity or oxygen levels. These shared solutions arise because life faces similar challenges across very different environments. When evolutionary pressures align, biology often converges upon the same molecular strategies. These parallels show that even without shared ancestry, there are only so many ways to solve certain problems. The genome rails at efficient designs through independent innovation. DNA can create biological innovations through rare mutational events. Most mutations in DNA have little effect, either because they change nothing important or because cells repair the alteration quickly. However, on rare occasions, a mutation appears that alters an instruction in a way that opens entirely new biological possibilities.
These unusual events can restructure how a protein folds, alter how a gene responds to signals, or create a new regulatory pattern that affects dozens of processes. Such rare alterations may pave the way for new traits, whether in behavior, physiology, or ecological adaptation. Although most organisms carry vast stretches of stable and conserved sequences, these infrequent shifts act as spark points for evolutionary innovation. They show that major changes do not always require sweeping modifications. Sometimes a single unexpected alteration launches a new direction that future generations refine. Life's diversity owes much to these rare sparks that appear quietly within the genome and radiate outward across future lineages. Some genes can reawaken after long periods of silence. In many genomes, certain genes become inactive for long spans of evolutionary time. They remain intact but unused, carried quietly from one generation to the next. Under the right combination of environmental conditions or molecular signals, these silent genes can awaken again, revealing their functions long after their activity first faded. Some may offer advantages only in particular climates or ecological contexts. Others may restore traits that vanished but become relevant once more as environments change.
Their reactivation provides an additional layer of flexibility, allowing lineages to draw upon stored potential rather than relying solely on newly evolved traits. The presence of these silent yet functional sequences reminds us that the genome carries a quiet reservoir of possibilities, instructions resting patiently, waiting for the right moment to guide survival. They sit in the background like unlit lanterns, ready to glow when the world calls for them. And in their stillness, they offer hints of something even larger. DNA may hold clues to life beyond Earth. Even though no living organisms have yet been found elsewhere, the structure and behavior of DNA reveal universal principles, patterns so elegant and adaptable that they may echo through worlds far from our own. The molecule's ability to store information, copy itself with reliability, and adapt through variation suggests a successful blueprint for organizing living systems. When scientists analyze the chemistry of DNA, they find patterns that could also arise in environments far beyond Earth, provided certain conditions are present. Some of the earliest features of the molecule reflect ancient adaptations that may parallel the challenges faced by life forming in distant oceans, hidden subsurface environments, or atmospheres rich in unfamiliar compounds. While life elsewhere might use different molecules, the strategies encoded within DNA offer insights into how information could be preserved, transmitted, and diversified in alien settings. By studying these strategies, researchers gain a deeper understanding of what life could look like on other worlds and how to recognize it if it appears. As we drift to the end of this quiet journey, let the thought settle that inside every living being is a world of patterns and stories, written in threads too small to see, yet vast enough to hold the history of life itself. DNA is silent, but it shapes everything. It guides the growth of forests, the rhythm of oceans, the breath of animals, and the paths of our own bodies as they learn, change, and respond to the world. There is something comforting in knowing that within every cell is a map of connection, a reminder that life is linked by shared instructions that echo across ages. As you sink into rest, let these thoughts become soft. Let them drift outward like ripples fading across still water. If you enjoyed this gentle journey, I invite you to like, subscribe, or share a thought below. It helps others find their way here, one sleepy soul at a time. But for now, let your breath slow, let your eyes grow heavy, let your thoughts unwind into quiet. Good night.